Thank you. Thank you for the kind invitation, Mani and Fernando and Raul. And congrats to the 10th anniversary. I never missed any meeting here, and I'm looking forward to the next 10 years or longer, I hope. After this highly scientific presentation we heard in the last 10 minutes, I come to a really practical topic, just to show bacteria during operation, laparoscopic operations and open operations, by their autofluorescence if we use a special light source. We all know about autofluorescence, but about fluorescence, but autofluorescence is not so well known in surgery. That means any background fluorescence that occurs in cells and tissues without any dye. It's just autofluorescence. It's primarily caused by metabolites and cellular structures that serve as fluorophobes, natural within the cell. We know this from plant in biology. This is a nice picture from Mr. Donaldson with autofluorescence in plants. It's well known in biology. Very nice pictures. And we have in nature a lot of common autofluorescent molecules. In mammalian cells, we know this from our enzymes, NHDA, collagen, elastin, and so on. They are all autofluorescent molecules. Interesting also tryptophan and lipofuscins, flavins, and porphyrins. And in bacteria, we know this from chlorophyll and bacteriochlorophyll, which are autofluorescent molecules, and in plant cells as well, chlorophyll. There's a special mechanism we know from fluorescence. You have a special wavelength for excitation, which is always lower than the emission wavelengths, and specific for each molecule. If you take NHDH in cytoplasm, for example, we have an absorption maximum with 335 to 350 nanometers, and the emission peak with 440 to 470 nanometers. That means if you want to make NHDA fluorescent, then you should have a light source with a wavelength of 335 to 350. And you need a filter with this wavelength of 440 to 470. The autofluorescence, the amount, is tissue or cell dependent. Interesting, in our own body, we have lipofuscin, an aging pigment, which is more and more when we get older, and it accumulates in lysosomes and in these long-living cells. And if you use autofluorescence, we can detect the age of our cells by measuring the lipofuscin. Very interesting. In clinical use, there are a lot of publications about identification of oral malignant and pre-malignant lesions in the throat, for example. And well known, it's for parathyroids. We heard a lot of presentations today. You see on the right side a publication of Sang Li with the presentation of adenoma with autofluorescence and multi-gland disease. Everything is shining in these glands with autofluorescence. In ophthalmology, it's widely used for the detection of diabetic retinopathy in the fundus. It's well known for a long time, as well as ICG in ophthalmology. And interesting, there's a lifetime accumulation as a biomarker. When you're a lot in the sunshine, in the open sun without sunglasses, and you don't take care of your eyes, you will have more and more metabolites which show autofluorescence of the retina, and your eyes are older. You can measure this as well. And the right lower picture is, I didn't know before preparation of this presentation, it's a skin autofluorescence we all have, and it predicts type 2 diabetes mellitus. It detects heart disease and mortality. It's published in Diabetologia in 2018, the skin autofluorescence. I have two examples of modern devices you can use to see autofluorescence. It's FlowBeam LX. It's a nice instrument for open surgery. It's handheld, and you can use it for autofluorescence and for ICG detection as well. And the other is Molecule Light IX. I like very much. It's like a mobile, it's handheld. I used it for a couple of years in diabetic feet care. When you make a wound control, you can see the bacteria in the edges of the wound. You can make a scrub at these areas. identification of bacteria. And this device is built with the exciting wavelengths of 405 nanometers and has a dual band filter with 500 to 545 and 600 to 665 nanometers. And you see how it works. You see on the left side the emission of violet excitation light. And then you get signals back from the skin, green and red and cyan. And normally a lot of bacteria have a red fluorescence, autofluorescence, and for example, Pseudomonas aeruginosa has a cyan fluorescence, and you can sometimes see by the color which bacteria are there, gram-positive, gram-negative, aerobic, anaerobic, and it's really fascinating if you see the next slide. For example, this was a wound control. You see red fluorescence of Staphylococcus hominis at the edges of this wound, and another wound, this cyan fluorescence for Pseudomonas aeruginosa. We always take a sample to the microbiology to see the bacteria, and this is a result. And there's a long list. This list is shortened by 50%, a long list of bacteria which show autofluorescence, mainly red fluorescence. You see the well-known bacteria like Staphylococcus aureus, Epidermidis, Capitis, and so on. For visceral surgeons, very important, Escherichia coli and Enterobacter cloacae. There are a lot of publications, wound care seen in a new light. For example, to see bacteria in chronic wounds, to find pathogenic bacteria in wounds, or to optimize treatment in Pseudomonas aeruginosa. And the conclusion by Molecular Light was that it can focus the surgeons, the clinicians, on potentially harmful levels of bacteria with the guidance of fluorescence imaging. Very interesting, I think, and very easy to use. This is literature from yesterday. If you search for autofluorescence, you find 15,000 results, but mainly biology with plants. If you add surgery, you have 2,500 results, mainly clear as parasaurids. And if you add laparoscopy, you only find 19 results worldwide, and they are only about endometriosis, like we saw before. Very interesting. Great that you are here. Same topic, a little bit. And if you add bacteria, we have 1,000 results. They are concerning an open surgery like biofilm detection in chronic wounds, pathogenic bacteria. On the other hand, ICG, our main topic, for more than 4,000 results concerning colorectal, gallbladder, and liver. How to do? What do we need to have laparoscopic autofluorescence? There's a big problem. When I started with ICG in year 2008, long ago, we had to use two optics. We had one normal optic and one optic with a special ICG filter, and we had to switch in the operation one optic then the other to see ICG and so on. Nobody liked this. For many years now we have this nice optic with ICG near infrared. And if for 2 years now we have this Rubina on the right side, it's inbuilt 3D, 4K, ICG and near infrared. It's really perfect. But you can't see autofluorescence. And for autofluorescence you need a special dual band filter, but there's no approved device on the market to use it. And therefore I had a problem. I cannot put a not approved camera into a body to do a laparoscopy. Therefore the little trick was I took a sample of the free fluid in appendix perforation outside of the body and then imagined by autofluorescence. And this was really a prototype to combine a special optic with a special filter. And this was the prototype. I had it for 3 months. I have to give it back now. But the light source, sure, like in Molecular Light X, is 405 nanometers for the excitation. And then we had a special dual band filter we could clip on the laparoscope outside the body with these two peaks, 402 to 440 and 603 to 655, similar to the Molecular Light X, where we know that it was working. And our first test was where we made a lab test. These are samples from the laparoscopy, and we put it in these agar plates and incubated it in my office for 48 hours, and then they make the test with this light source, and we could show this is E. coli. In this example, there are only some little red spots to be seen. The others, I think, are already dead, this bacteria. But in this case, this was a perforated sigmoid diverticulitis. We could show Enterococcus faecalis, and you see a lot of red dots in these agar plates, and this is the red autofluorescence of Enterococcus faecalis. And to show something, for practical use in open surgery, there was an abscess of the abdominal wall. It's clear there's pus, and you have to make an excision and remove the pus. And on the upper right picture, I think the wound seems to be quite clean. And then you make autofluorescence, and you see in the middle of the wound and some edges there are still autofluorescence by Staphylococcus aureus. And you can then remove this till it's clean, and you have this on your instrument. The bacteria you can show in the camera, there are the bacteria, and you can then show that the wound is clean. Another interesting case, 6 weeks of therapy in the clinic and 20 operations, a patient from last month with Fournier's gangrene. This was the second revision. You see still that the wound is with liver and tissue and biofilm, something like that, but you can't see it exactly. Then we use the laparoscope with the dual band filter, and you can exactly see where the bacteria are located in this wound. And it was very funny, there's a little spot on the surgeon's glove, and you can see that this spot contains bacteria. And then you can make a revision and washing, and you can see at the end the wound is clean and there are no bacteria. What's the clinical role of this relative new thing? It's, I think, the only possibility to visualize bacteria live during the operations. You all know, I had some situations before. I want to do a TAP procedure with a implant, with a mesh implant, and I look into the abdomen and I see creamy fluid like pus. I took a probe, a sample, stopped the operation, no mesh implant. It was Friday, the result came on Tuesday, it was sterile. Then Wednesday, next operation, I implanted the mesh. And I have a lot of patients, I think 1 a month, with unknown fluid in the abdomen, and you don't know whether it's pus or not. And the possible indication and advantages are detection of peritonitis. Sometimes, or we do it quite often, when we have a 4-quadrant peritonitis, we do a second look on the second day to make another washing. And that was what I was waiting for, but I couldn't do now because it's not approved, that you put in the laparoscope in the second look laparoscopy, and you can see in all quadrants whether there are still bacteria, there's fluid behind the liver or behind the spleen, and you can see whether it's clean or if there are still bacteria, and you are forced to do a second look or a third look. And that's the same for laparotomy. In laparotomy, it's no problem. In open surgery, you can use this camera because you don't bring it into the abdomen. Therefore, it's now easy for open surgery. And we had for a long time this molecular X, what we use for diabetic feet, but as well in open surgery for the abdomen to watch whether there are bacteria or not. If you do a re-laparoscopy, for example, if you are afraid of an anastomotic leak on the second or third day, you make a re-laparoscopy, and that could also help us to see whether there are bacteria around the anastomosis, there shouldn't be any, or if it's clean. For really severe patients on the ICU, you have fluid in the drains. You could put a sample or put the drain back and put it to the cameras to see whether there are bacteria in this drain or not. That could lead you to indication or no indication for reoperation. Only problem is that we, up to now, don't have any optics that are approved for laparoscopy, but we hope that the industry will help us to build such an optic, like we saw this in ICG. First with a filter, then inbuilt. I think it's not such a problem. And the light source is also no problem with 405 nanometers. Last year I stopped with this slide, routine use of ICG in colorectal surgery, and my conclusion was see better, treat better. And I can transfer this to this year for clinical role of autofluorescence, the same with see better, treat better. And thank you so much. We are sitting in the green bar.